Hello and welcome to Unit 1.3 Lipids. This is a short revision video. If you would like a longer, more detailed version of this video, then check the top link in the description. You can also find other resources such as flashcards, detailed notes and revision websites in the description. I will now go over the key points of this unit. Let's recap Unit 1.3. Lipids are a mixed group of hydrophobic compounds composed of the elements carbon, hydrogen and oxygen. The main complex lipid types are triglycerides and phospholipids. Both contain glycerol and fatty acids, except in a phospholipid, one fatty acid is replaced by a phosphate group. Fatty acids. Fatty acids have a carboxyl group and a hydrocarbon chain. All fatty acids have the same basic structure, but the hydrocarbon tail varies in length and saturation. They may have no double carbon bonds in the chain, making them saturated and straight chains, or they may have one, monounsaturated, or several, polyunsaturated, double carbon bonds, making them unsaturated. This causes kinks, bends in the chains. This lowers the melting point. Triglycerides. Triglycerides are commonly called fats or oils. They are made of one molecule of glycerol and three fatty acids attached to it. Glycerol is a small, 3-carbon molecule with 3 alcohol groups. They form through a condensation reaction where 3 fatty acids form ester bonds with the glycerol molecule, producing 3 molecules of water. Triglycerides are insoluble in water. They are used for storage, insulation and protection in fatty tissue, or adipose tissue, found under the skin, subcutaneous, or surrounding organs. They yield more energy per unit mass than other compounds, so are good for energy storage. Finally, phospholipids. These are similar to triglycerides, but a phosphate group in place of one fatty acid chain. The phosphate head is hydrophilic, loves water, and polar. It is therefore water-soluble. The two nonpolar hydrophobic tails, the fatty acid chains, repel water. Phospholipids are the main components of the cell membranes, also known as plasma membranes. They make up the phospholipid bilayer. The hydrophilic heads face the water and the hydrophobic tails face each other. This traps a compartment of water in the middle, separated from the external water by the hydrophobic sphere. You can test for lipids using the emulsion test. This involves mixing the sample with ethanol. This should dissolve any lipids, as they are insoluble in water but are soluble in alcohol. Then, decant the ethanol into water. A while emulsion will be visible if lipids are present. Hydrophobic means water-fearing, nonpolar molecules that are lipid-soluble. Fatty acids are hydrophobic. Hydrophilic means water-loving, polar molecules, water-soluble, but not soluble in lipids, such as the phosphate heads on phospholipids. Thank you for watching Phreology Revision.